So the
Yeah,
floor is yours.
thanks a lot. Um, so welcome back to the session. First of all, my heartfelt thanks to the organizers for for this awesome invitation. I really appreciate it. And this has been a really amazing conference. I've learned so much over the past week. Uh, and Tokyo itself is amazing. So a little bit maybe about my whereabouts, where my lab is. So I come from this very flat but also very beautiful country called Netherlands, and my lab is actually in Wageningen University at the Department of Physical Chemistry and Soft Matter. It's a very diverse group of people. We study from polymer materials and coatings to complex fluids, and I'm of course interested more in the self-assembly of biomolecules. Even within my group, we study a lot of different topics, from protein condensates to vesicles, membranes, and we are also doing some biosensing. And we use a variety of tools to do this. Uh, so microscopy, spectroscopy, uh, lots of physical chemistry tools, but also microfluidics is something that we do a lot in the lab. So, I was thinking of how to introduce this topic, and Tokyo itself provided the answer. So, this is a very nice projection on the Tokyo Metropolitan Building. And basically, what they are doing is self assembling things to create life, right? So, I mean, they make it sound easy, and we know that it's not. But the essence is the same. And even I would say, if you look at very simple multicellular organisms, this is a fantastic organism called Trichoplax adherens. It has only like 6 cell types or so. But still, this is a very, very complex system to study, especially if you want to understand the molecular interactions. Uh, I would say even single cell bacteria basically are very, very complex. So for me, if I want to understand really the molecular mechanisms behind living systems, I like to go to the molecular level, to the protein level, to really just understand how they can interact with each other and so on. So since we are talking about life, death, non-life a lot over the past 2 days, I thought of bringing this up. Maybe it's a little bit of a bold statement, but defining life is extremely hard, and I do wonder from time to time the usefulness of that in the context of. Uh, I don't think life as a very binary thing, but more of a continuum. You can think of a degree of lifeness basically to your systems, because it really depends on you where you want to draw the line, right? Would you call a self-sustaining energy production entity alive, or you would call it only when it is capable of evolution? And I think there is a lot of gray area basically in between. And as an experimentalist, for me, the most useful thing is trying to build it. So, and seeing where we can actually go, right? Because we are anyways taking very, very tiny steps here. So, indeed, the question is, can we build life from scratch? Can we start with individual dead or non-living components and try to assemble it into something that mimics key characteristics of life? That's what we want to do. And I want to come back to this beautiful book by Erwin Schrödinger, What is Life? And I'm sure many of you have read it. It's a fascinating book with many interesting thoughts. Uh, and of course, he's talking about how can physical and chemical laws basically govern the living systems. And I want to point out a couple of things that are, I think, relevant to this talk. The first is, he talks about living systems are basically made up of a large ensemble of molecules that make it uh, that can be then controlled through statistical laws. And the other thing is this out of equilibrium conditions, right? So out of equilibrium is extremely important for living cells to keep it, away from death, basically. Uh, and both these conditions, I would say, are met through a clear pre-requirement for life as we know it, and which is basically confinement, right? So all the life that we know today is actually confined. So confinement produces this kind of a collection of molecules that can allow it to interact with each other, and it allows for all sorts of non-equilibrium gradients, like pH gradients and osmotic and whatnot. So confinement is indeed actually what we are fascinated with and what my talk is all about. It's all about confinements, how we can make them in a controlled manner, how we can deform them, how we can basically subcompartmentalize these things. So I'm going to tell you a few short stories. Uh, so yeah, that's kind of the essence of the talk, right? How can you compartmentalize systems? So a little bit about these vesicles, because I will show an example of each of them. You can think about all sorts of vesicles to experiment with. You can start with very simple ones like single emulsions, which is basically water in oil or oil in water emulsions. You can go to double emulsions, which is a more complicated system with water in oil in water kind of emulsion. And of course, you can go to liposomes, which are basically aqueous confinement surrounded by a lipid bilayer. And I would say, as you go from left to right, they become more and more biomimetic, but they also become, I would say, a little bit more difficult to make. The other sort of confinement I want to also refer to is actually not membranous confinement, but a phase separation. Now, I think you have heard a lot of talks about the biomolecular condensates, but I also want to refer to both sorts of phase separations, the segregative phase separation and the associative one. So in the segregative phase separation, the basically you end up with two polymer-rich phases, right? Each having a certain kind of polymer, and I will show a lot about dextran peg polyethylene glycol system. The associative phase separation, normally, I mean, it's also called coacervation or more generally as condensation, where both the species basically come together to form a polymer-rich environment in equilibrium with a dilute phase. And we are going to make use of all of these systems. So I see synthetic cells as basically playing Lego with biomolecules. And of course, you want to complexify your system and try to see, okay, can we get something interesting? But I also like to look at it the other way around. How simple can we go and still get something interesting, something emergent? And I want to give one example of that. So this is an experiment that involves only two components. There is nothing else in this experiment. Yeah, it's water and oil. It's a very simple experiment actually. What you have is we take a glass slide, put a water droplet, and we make micron size oil droplets, decanol here, and basically put them on top of our water droplet. That's it, nothing else. So here we've also put a fluorescent dye, but it also works without fluorescent dye. So it's really a two-component system. And let's just simply watch what happens. So you saw that burst, right? And it's spontaneous, it just happens. And not only it happens once, it can happen again. And this just keeps on continuing. Once my student, I think, recorded 40 such events. So this is something quite fascinating, and we were like, we need to figure this out. I don't want to go into too many details, but I want to show you another movie because I think it really shows the mechanism behind this. Oh, by the way, here is a time lapse. So you see again, right? I mean, there is a collection of droplets basically. Um, and oh, yes, perfect. Thanks. Yeah. Oh, this looks much better. And you basically get this first pop, as we call it, and then the droplets again start to diffuse around, and then another droplet basically pops, and you get another redistribution event. And what is actually happening is stochastically, a decanol droplet basically enters the air-water interface and starts producing this beautiful Marangani flow. Maybe I should go back once to show you again this movie. Yeah. So it enters the surface and it produces the Marangani flow, which repels the other droplets basically. But at the same time, there is also evaporative flux of this oil into the air. It is small, but for a very small droplet, it is significant. So what you end up basically is a continuous reaction. The droplet enters the surface. Over time, it evaporates, it simply vanishes, and then another droplet pops, and this cycle just basically continues. And I think this beautifully illustrates how I mean, you cannot go more minimal than two components. With one component, nothing is going to happen probably. But even with two components, how we can get a spontaneous reaction that continues. Um, all right. what can you do with it? So I want to show again a couple of enzymes. For example, here I show a video where 5 of these droplets have popped and gone to the sides of the water droplet. And what they basically do is each of them has a Marangoni field, right? And basically that's why they keep all the rest of the droplets basically in the center. So it's like similar to optical tweezers. We call it Marangoni tweezers basically, right? Uh, Another cool thing you could do is if you have a collection of such droplets, all heterogeneous and so on, this popping can provide internal energy for the redistribution. So you can see right at the start, it was all, it was mixed population, but you provide some internal energy, and then suddenly the large droplets, which have a higher buoyant force, can go into the middle, and the other droplets basically can surround it, forming almost like a tissue-like structure. All right. So now I want to kind of go more complex and also enter the microfluidics regime. So what we want to do now is compartmentalizing our containers using biomolecular condensates. And here for the container, we are going towards double emulsion. So these are water in oil in water emulsions. Yeah. Now, this is something that living cells do a lot, right? I mean, they do it on a very high level, but we simply want to start thinking about how we can do this in our case, and in a triggered manner, right? In a very controlled manner, exactly when we want. And if you think about the basic parameters you want to basically tweak, I would think of these four. pH, right? It's a very basic parameter. Temperature, salt, so osmotic strength, and the concentration of the component itself. That could be nice. So now there is a family called elastin-like polypeptides, and you are probably aware of this. These are synthetic mimics of elastin. These are basically pentapeptide chains of GXGVP. X is any guest residue here. So ELPs is a very widely studied family of proteins that can undergo phase separation under temperature. But we recently designed with a collaboration from the lab next doors from Renko, we actually designed a family of pH responsive ELPs which respond to all the four triggers. So as the pH goes down at a particular pH, we get a transition, and you can see an example here. But they are also still responsive to temperature, salt, and its own concentration, of course. So this is a very nice system, we think, to basically play with condensation upon trigger. So microfluidics. We love microfluidics for a couple of reasons. It's cheap. It's rapid prototyping, right? So we can change our device designs basically a week max. But I mean, over a few days, we can tweak our designs. We can work with low volumes. 100 microliter is actually quite a lot. If you want, you could also work with 10, 20 microliter of your precious sample. It's a high throughput method. Just a bunch of devices I made in a week, maybe. I don't know. And I mean, of course, you know about microfluidics, but I think this really conveys the essence of microfluidics, that you have turbulent flow in a bulk process, while in microfluidics, everything becomes laminar, right? The inertia kind of goes away, and you can really play with this, for example, for blowing bubbles. So this is a typical double emulsion production that I'm showing you, right? So the proteins, the ELPs, the pre's, are getting encapsulated in the inner aqueous phase. They're getting pinched off by the outer aqueous to produce these beautiful double emulsions. And you can, I think, already sense the control over it. You can make a lot of them, and they're all nicely monodispersed, and you can control the size and so on. Now, if we take the double emulsion, because we also wanted to show basically the versatility of our double emulsion system. So you can take our double emulsions filled with these proteins, and if you basically acidify the environment outside, the pH will equilibrate over time, and you will get this beautiful condensation process, and you get one condensate in each double emulsion. You can reverse the process, and they go away. Yeah, you can do the same with temperature. You can simply put your double emulsions on a hot plate. They will undergo phase separation. You can go there, take it back, and repeat the process, and so on. We wanted to have a bit more control, and here now the example is with the osmolytes. So if you take these double emulsions and basically just let the buffer outside evaporate, you create a hypotonic condition. The double emulsions will shrink, the salt and the ELP concentration will increase, and you will get phase separation. But now we've decided to do this in microfluidics. through basically trapping these double emulsions in physical traps and really changing the outside environment, in a rapid manner. So this is kind of a image of these traps with double emulsions trapped. Yeah. And now I want to show a movie which I think goes over 7 hours. Not now, but it's a sped up movie. So just focusing on two double emulsions trapped here. And basically when you switch on the hypertonic trigger, you will start seeing condensation in the double emulsions. You see that tiny dot here. You switch it off, it goes away. You switch it on, it comes back, and you can basically keep on doing this until you get bored. So I think it's a very nice system to do this kind of condensation in a very, very controlled manner. Uh, and here is a time lapse showing, I think, 7 cycles of condensation, dissolution, condensation, dissolution. So I think these double emulsions are a very nice system. They are very easy to make, actually. If you want to just study condensation reactions in tiny cell-like compartments, and it doesn't have to be condensation only, it can be something else that you would want to study. The double emulsions are extremely stable from pH 1 to 13, temperature of, you can keep it in the fridge for weeks until 50 degrees. So it's a very, very robust confinement. That makes me go to the next story. Now we are making it more complex. So double emulsions are great, but only if you don't want or you are okay without any membrane interactions, right? So there is no real membrane to it. If you want actual membrane, you want to work with liposomes, and that's where this story is. So we were thinking, can we basically use both associative and segregative phase separation together to make more and more complex synthetic cells, more hierarchically assembled synthetic cells? Is that my time basically? Okay. Okay. Oh, perfect. Yeah. So, and of course, associative phase separation is kind of akin to the biomolecular condensates, and we have lots of examples of that in the cells. Segregative phase separation is a bit of it's an interesting way. I mean, we think that it's akin to sort of molecular crowding inside the cells because there are lots of polymers, and it's quite possible, I would say, that there is some sort of segregative separation between these polymers as well. So both these things must be basically taking place in the cells, and that's what we wanted to do. So we work with this 4-component system. The PEG and DEX can do the SPS, and ATP and polylysine, which is basically negatively charged component and a positively charged one, can do the APS, so the associative. Yeah. Now, first things first. Basically, for all the experimental conditions that we used in this story, we looked at the binodal of the PEG DEX, basically where the PEG and the dextran can undergo phase separation, and we see a very stable binodal regime. Basically, above this red curve, you have phase separation. And now the interesting thing is that we have chosen the components in such a way that one of the components, polylysine, likes to go into the dextran phase, while the ATP doesn't really care. ATP doesn't really partition into any phase. It's just there. Yeah. But now something really interesting happens when you put all the 4 components together. What you get is basically dextran droplets in PEG, which is very normal. But then, because polylysine goes into the DEX phase, it also coacervates in the DEX phase. So what you have already is a hierarchical assembly. You have your coacervates in your DEX droplet, which is in the PEG solution. Yeah. And this is simply happening because polylysine prefers to be in one phase. And you can see that very well here. They really physically restrict the coacervates. So we are just putting some flow on this on this slide, and the coacervates simply keep on rotating within their dextran phases. They cannot move out. Basically, they are now confined. But not just they are physically restricted. If you they are also molecularly restricted. So if we do FRAP on them, so fluorescence recovery after photobleaching, in a normal scenario where you don't have this, by the way, we call this SMCs, yeah? So another short form, sorry about that. So SPS-based membraneless confinement, so SMCs. So without the SMC, you just simply have a full recovery of your coacervate. That makes sense. It is in equilibrium with the dilute phase, and it's going to recover. But if you do a FRAP on a condensate that is inside your SMC, it actually does not recover. And that is because it is simply isolated completely from its environment. So it has very little molecular access to its other components. So it's truly confined. And this could be a nice way to make use of such tiny confinements within cells, within synthetic cells. Okay, so if you do it in double emulsions, right, this is not true membrane, there is no membrane affinity. What you simply get is the same as you saw in bulk. You get basically a dextran phase and inside a condensate. Yeah. And now things become interesting when you start doing it in liposomes. But for that, I want to spend a couple of slides on this liposome production method. This is something that I developed during my postdoc, and we call it OLA, for octanol assisted liposome assembly. And this is basically a very similar blowing process that I showed earlier, right? So we blow these bubbles using octanol as the lipid solvent, and you can see these tiny pockets. Here is the lipid channel, here is the protein that we encapsulate, and this tiny pocket basically go away within minutes to give us this. Yeah. So we get a nice sea of liposomes to play with, and a few more characterization slides on this. So first of all, these liposomes are unilamellar, meaning it has one single bilayer. We have done this with alpha hemolysin test, which is a pore that can basically let small molecules diffuse. So if you have alpha hemolysin, the fluorophore will go out. If not, it will stay. We have also done this diethionate bleaching assay where you basically make NBDP based liposomes. And if you add diethionate outside, it will quench only the outer leaflet. So the fluorescence, the fluorescence intensity should basically go to half. And this is basically what we see. Yeah. Also, the lipid diffusion is very similar to what you get with electroformed vesicles, for example. So if you do FRAP, it recovers, and the lateral diffusion is very close to the electroformed vesicles in terms of presence of surfactant and absence of surfactants. And finally, also the lipid composition is maintained. So what you put inside is what you get out in your vesicle. So here is a simple confocal microscopy-based measurement where you increase the amount of fluorescent lipids, and you will get more and more fluorescent liposomes basically. And you can even change these are from basically other groups who are also using OLA. This is a latest paper where they actually on the fly change the lipid composition. Basically, instead of one single channel of lipids, they have two, and they can change the lipid composition as you go on making the liposome, so as to make different compositions. All right, back to the story of the APS SPS. So now, here is a liposome, red is membrane, and we have put all the four components inside, right? The APS components and SPS components, and now we trigger both of them through an hyperosmotic shock as well as a pH shock. Yeah, and let's see what happens. So at the start, you see the liposome just diffusing around, but as the shock comes into play, you start seeing this transformation of the liposome into sort of a flower-like shape. You start seeing these dextran domains. Sorry, the movie quality is not that great, but the liposome is moving around. It's a bit difficult, but you can, I think, see the shape change clearly, and you can even see the coacervate now into that dex domain. Yeah. You can, if you look at the heat map, basically you get these dex domains, and the polylysin intensity is basically in that. I think it looks much better like this, right? So here we have our flower-shaped liposome now. We call these petals of dextran, and within those petals, actually, we have these buds of the coacervate. So we have a basically double-layered organization. We have a segregative phase separation helping associative phase separation to further get restricted. And here are the basically cross sections showing again beautifully the dextran domains, and in some of which are basically our coacervates nicely there. And they really restrict again, right? So the diffusion, if you track their diffusion, it's very different from what you will have without any such petals. The diffusion coefficients are very different. It's actually very akin to what is known as a choral diffusion, where you have this little restricted sites. and the diffusion is almost basically half of what you would get with without our SMCs. Okay, so I think it's a very nice strategy to use also this associative phase separation as an orthogonal strategy to the segregative phase separation and bring about hierarchical assemblies in synthetic cells. And it can be very useful for many of the reactions maybe even you have been using, right, with synthesizing all sorts of things in a compartmentalized manner. Okay, I come to the last part, and we use a lot of microfluidics and so on, but we have also been always thinking new ways of doing things, and we were like, okay, can we spray synthetic cells? And when I say spray, I mean electrospray. So electrospray, is a very, it's a soft ionization technique that revolutionized basically mass spectroscopy. And here we build a very simple electrospray setup. We have a syringe with an electrode. We have the ring electrode beneath it, and we basically spray the solution. This is actually a movie. It does not move too much because this is a Taylor cone, which is very stable. And in this stable regime, actually, you can shoot out droplets. And the droplets basically simply fall into the petri dish. You can see it here. And our idea was actually, could we use small unilamellar vesicles that we put into our dextran phase, and would this electric field somehow restructure them on the interface and make some sort of liposomes or some other structures? And we were in for a surprise a bit, because they did produce structures, but quite different from what you would expect from microfluidic process or liposomes. These are clearly not liposomes, but they are still interesting, I think. So what you get is kind of this irregular lipid nebulae around our dextran droplets. They make it very, very stable, though, so they can stay for weeks. And in fact, if you look after a week, you see two interesting things. First of all these dextran droplets start to kind of associate with each other to form these kind of networks. And if you observe at the bottom of the dish, you start seeing some very, very round droplets, which nothing like these irregular shapes. If you look at them carefully, you actually see a very thin film of lipid around it, which almost seems like it's a lipid bilayer, basically. So we think it's really over time that some of these droplets can basically sink, leave out all the excess lipids basically in there, which can also be maybe useful, to have some, instead of lipid synthesis, maybe you can use this kind of lipids as a reservoir or so on. So, another interesting way of making synthetic cells, and there might be some potential we will explore further. So I showed you 4 stories, right, about how you can go very simple just with 2 components and still make something, some emergent systems. Then we went to the double emulsions and condensates triggered by pH or temperature and salts. Then we talked about the associative and segregative phase separation inside liposomes. And finally, I showed you a new method of making some weird synthetic cells. Now, talking about all this, it really made me, look back a bit of how it all started for me. So it was in 2016 when I developed Ola to make these nice liposomes, and actually we divided liposomes in a very very crude brute force manner by basically bombarding them against a splitter, basically. So it's a very mechanical division, but it still works. We actually also tried to grow liposomes using membrane tension mediated SUV fusion. And then in the late, by the way, then we also use liposomes for drug screening and so on in collaboration. And it was in 2019 that I really became interested in condensates, to start making condensates in liposome structures. And then that really started a new phase for me, because then I started my own lab, starting with condensate membrane interactions. This was still postdoc work, basically. But then going towards, I did not talk about this, but we also made all sorts of other things like actinosomes, which are made up of actin, but not using it as a cytoskeleton, but as a boundary protein. We started using biosensing. So these are quite different projects, but they can still be useful for synthetic cells, I think. And then we started going into this stimuli responsive condensates. You show the flowers, of course. The protein transitions really become a big part of my lab, and we are working a lot with that, not just in the context of synthetic cells, but other things as well. And now what we are trying to do is actually trying to drive enzymatic chemical reactions in those condensates. So we are now really going towards the chemistry as well with all the tools that we have. We really want to think of these condensates now as bioreactors and try to drive enzymatic reactions. Uh, yes, I mean, oh, this is all thanks to my lab. Some really enthusiastic people. We are a small group, but a very energetic and creative team. And I'm very thankful to these guys for doing all the amazing work, and of course also the funding. Uh, Along with synthetic cells, we do other things because we do microfluidics a lot and do all sorts of emulsions. For example, we play with Pickering emulsions. These are particle stabilized emulsions, quite different from surfactant stabilized emulsions, but also very interesting. We have been working with natural proteins and their phase separation potential, for example, in tick bioadhesion. Uh, and this liquid crystal biosensing is something quite recent that I have gone into, and these are also emulsions that we are using to sense, again, amphiphiles and lipids. And I mean, I think this photo nicely summarizes that I had a lot of fun here, not just at the conference, but also outside. So thanks a lot again for the hospitality and for the attention, and I'm ready to take questions. Thank you.
Okay, so we have plenty of time for questions.
So thank you for very beautiful pictures and nice talks. So I have a question about the networking vesicles with, how to say, fluffy, something. So in this case, the green coarserate is completely, how to say, closed by the lipid membrane or not. So can we, how to say, supply something into this green coarserate things?
Yeah, no, I think it's a good question. First of all, this is not a coarserate now.
Oh, yeah.
This is now dextran droplet.
Yeah.
So strictly speaking, it's not a coarserate. But I do think that it is possible to supply things in there because I don't think these are completely closed structures, right? These are, I think, just agglomerates of those SUVs. We have not done it, but would be nice to see. Yeah. And also the fact that they kind of associate with each other, you could think about some sort of communication between them. Yeah.
Yeah, thank you.
Thank you very much. Very nice talk. So I have a question about this, the research. So the riposome itself is a very, very stable, right?
Yes.
And you succeeded to align this riposome around the liquid-to-liquid phase separation interface, right? And then give the membrane fusion to make a lipid bilayer.
Right? Are you talking about the flower-shaped liposomes?
A very thin, red circle after giving the
here.
Yes, this one. Yes. The is it true that the thin red circle is a lipid bilayer?
We don't know, to be frank.
We don't know.
Yeah, we could try to do it was a bit difficult to do these measurements because they have now sunk to the bottom of the petri dish, and it was difficult to harvest.
You mean that only the vesicle have this thin layer, right?
Yeah, sorry, can you repeat?
You said the thunked vesicle has this red.
Yeah, the sunk vesicles, yes.
Only
the sunk vesicles, yes. So basically we get two populations. We get these floating networks that just remain on top, and then some of them, I mean, not it's a finite population that just sinks down, and it really has this extremely thin lipid layer. We have not checked for the bilayer.
My question is, what energy induced this thin circle, the red one?
Yeah, so I think the dex over time, you do have the density does the density is higher for these droplets. So from time to time, there are some of the structures which are not in very close association with these networks can actually fall down. And as they fall down, I mean, these SUVs are not intact SUVs anymore because they were electrosprayed, right? So this electric field has actually disturbed the SUV structure basically.
Sorry, last question. What is the repeat composition of this small vesicle?
Good question here. So this is DOPC, DOPG, DOPS, MPEG, DOP, and some fluorescent. And this we took from based on the literature, also based on their potential to fuse with each other and so on. Yeah.
Good. Oh, thanks. Beautiful. Yeah, I was wondering in your SMC budding membrane, that are the membranes phase separated? No,
No, in this
Okay.
particular case, no.
just by the very strong wetting surface.
Yeah,
right?
yeah,
Okay.
yeah, yeah.
In-
So
yeah.
no,
With by the decks, so wetting is induced by decks.
exactly. So the dextran always likes to weight the membrane compared to the peg, and that's why it also depends on the volume fraction of the dex and peg. But we choose it in such a way that the dex will weight the membrane and form these microdomains. And yeah, I mean, over time they will fuse. Yeah, so that is still a thing to work on,
Okay,
and
yeah.
that's where the lipid composition can come into play because if we can also phase separate the lipids along with the dextran, that's a great point. I think the coarsening is reduced a lot.
Oh,
So yeah, something to do for
yeah,
the
great. Thanks.
future. Yeah.
We have 2 more minutes.
Hi, Siddharth. Thank you for a nice talk. I had a question about the last example that you gave with the SUVs at the interface. Did you also try to optimize the SUV concentration in that experiment? Because it looked like you had quite a lot of lipid, way more than you need for a lipid bilayer.
I absolutely agree. We did try a little bit, not too much. We tried a few different concentrations. And indeed, if you go lower with the concentration, this kind of corona reduces a lot. But still, we don't get a very continuous distribution. We still get this kind of irregular things. But yeah, if you add more lipids, you get a really big nebula. Yeah.
Yeah, thank you.
Hey, I think we had plenty of questions, and so let's thank the speaker again.
Thank you.
Thank you.